Okay, we're exploring nephron loop physiology, but first we need to identify a little bit of anatomy here and remind ourselves of what's happened before we've gotten to the nephron loop. So you can see right here, I have a blood vessel called the afferent arteriole, bringing blood into the glomerulus. I will push some of this fluid out, and that will become the initial filtrate that we're running through the nephron. That blood vessel leaves as the efferent arteriole and becomes the peritubular capillary, which is running alongside the proximal convoluted tubule and the distal convoluted tubule. That peritubular capillary then descends into a series of blood vessels called the vasa recta that line up with the nephron loop, and then those blood vessels leave. They'll go to the interlobular veins and eventually to the renal veins and out of the kidneys. Also, just to identify what's already happened before we get to the nephron loop, here in the proximal convoluted tubule, that tubular fluid has lost 65% of the sodium that it contained back to the bloodstream, back to the body, and 65% of the water in the tubular fluid has also moved back to the bloodstream, back to the body. At this point, that 65% of sodium and water, that's our maximum capacity to move those two substances, and we're sort of in a bind, because if that's all we moved, our urine would be incredibly salty and very high in water content, and we'd be constantly losing these substances from the body in amounts greater than we would want to lose them. So we need a nephron loop that can gather more of that sodium and water back to the body than we can via the proximal convoluted tubule alone. We're going to use two shared principles in order to do this process of the nephron loop and regaining extra sodium and extra water. So let's talk about these principles, because this is really the most defining idea of the nephron loop. And the big word here that I want you to think of is countercurrent. The countercurrent flow of the nephron is going to allow us to do two things. The word countercurrent is implying the fact that the fluid in the nephron loop is flowing in this direction, while the blood is flowing through the vasa recta in the opposite direction. So the word countercurrent is referring to the opposite directions of the fluid flowing through the tubule and the fluid flowing through the vasa recta. We get two incredible capabilities with this countercurrent. The first one is something called countercurrent exchange. Countercurrent exchange is the idea that when two substances, and for us it's fluids, the tubular fluid and the blood in the bloodstream, when two substances are flowing in opposite directions, we get rapid and more efficient trading of material between the two substances. And so that countercurrent exchange is really referring to the fact that our materials move across quite quickly and efficiently when my fluids are flowing in two opposite directions. The other countercurrent idea that we're going to employ here is something called the countercurrent multiplier. The countercurrent multiplier is a slightly more complicated concept, but the gist of it is that it allows us to keep an incredibly high osmolarity deep medulla and a low osmolarity in my shallow medulla, and we're multiplying the effects of the movement of our substances. So the countercurrent multiplier refers to the great difference in osmolarity we can maintain between shallow medulla and deep medulla based upon this countercurrent flow. So let's walk through this countercurrent flow and see what's happening here and identify the substances that we're moving out in different locations. So as we run our tubular fluid through the proximal convoluted tubule, we identified that we moved 65% of sodium and 65% of water back into the body, and now this fluid, which is still at equal osmolarity to the rest of the body, is going to flow down this descending limb. Right here, I've identified that this descending limb is also sometimes called thin segment, and the thin segment has a key pair of characteristics that are very important that we want to identify. This thin segment is highly permeable to water, but impermeable to salts, which means that as we begin to flow down into the deeper medulla and the osmolarity is getting greater and greater and greater out in this tissue, the water molecules will move down their own concentration gradients, which means they will leave the tubular fluid, enter the tissue of the medulla, and eventually diffuse into the vasa recta blood vessels. So this is water moving down its concentration gradient. Why can we get more water to move here? Because we've actually created a water concentration gradient that was absent up above the medulla. So water is going to move down its concentration gradient. Although sources differ, McKinley tells us that we can move another 10% of the water. I'll go ahead and put this 65 up here from before. We can move another 10% of our water in by this mechanism. Now as we round the bend and start moving up the ascending limb, you can see that the features of the ascending limb have changed. The ascending limb is often called the thick segment, and the thick segment is impermeable to water, but permeable to salt, and salt is going to move down its concentration gradient. Now what does that mean for a salt concentration gradient? Well, we were very salty down here in the deep medulla, and of course very salty in the tubular fluid because we lost that water. But now as we move up out of the deep medulla, remember that the osmolarity is dropping again. And so as the osmolarity drops, salt moves down its concentration gradient. Sodium, I should say. Sodium ions move down their concentration gradient. And we recover about 25% of what had been in that original tubular fluid. At this point, we need to identify how it is that the deep medulla can continue to stay salty, because we've been adding water to it over here. Wouldn't that dilute this 1800 milliosmoles? It would, except for the fact that this blood in the vasa recta is going the opposite direction. So our countercurrent multiplier comes into effect. As this blood comes around the bend and starts running down the ascending limb, it picks up the salt and becomes saltier and saltier as it comes into the vasa recta. Comes into the deep medulla. It can then sort of exchange salts with the deep medulla, keep the deep medulla salty. As it ascends next to the descending limb of the nephron loop, it picks up water, but it is leaving the medulla, and so the water doesn't remain in the deep medulla, it's carried away. That's the beauty of the countercurrent multiplier. So just an aside I want to point out to you, because it somewhat applies to our nephron loop physiology, is something that's happening over here in our collecting duct. So if we remember that we are staying at 1800 milliosmoles in the deep medulla, we don't want to forget the fact that this collecting duct has to travel down through the deep medulla to get to the renal papilla and get out of the kidney. And so this collecting duct is running through a very high osmolarity environment, and this is why, as the collecting duct embeds little principal cells, that respond to antidiuretic hormone and embed more aquaporins in their membrane. Why embed more aquaporins? Why embed something that's just utilizing a concentration gradient? It's because we're back to that incredibly high solute deep medulla, and the water will move down its concentration gradient, leaving the collecting duct and moving back into blood vessels that are running next to the collecting duct back into the body. That's how we can regain more water via the principal cell aquaporins in the collecting duct.